Let's back to Keynote. So I think COVID is making everything interesting, including discussion of scientific papers. I think this week was very topical, specifically because you often hear various people claim that it's easier to follow the science on COVID. By using the definite article the implication is that there is one sort of standard, widely accepted truth and set of facts that everybody agrees on. And in some cases it's true, but in many cases it is a lot murkier than that. So today I'll begin by first circling back on what we discussed last week, which is, we spent a considerable amount of time talking about clinical trials, and specifically the ACCT1 trial, which was the trial ran in the spring. And the primary outcome of that trial was that remdesivir became, which if you recall is a direct acting antiviral drug that inhibits the action of RDRP. It's RNA-dependent RNA polymerase, was originally developed for Ebola, so it's a repurposed drug. And that trial was declared a success and led to the approval under the compassionate use by the FDA in May. In fact, I just saw the news today, I haven't confirmed it, that the FDA released a formal approval for remdesivir this week in the United States. I believe that's true, but I haven't checked the primary sources, so I could be wrong. And the primary outcome of that trial was that patients in the remdesivir group had a shorter time to recovery than patients in the placebo group. The effect was about 5 days median, and that was a highly statistically significant result. The important secondary outcome was that there really wasn't a significant effect on the longer term estimate of mortality. So what was shown here is that you had 11.4% people in the treatment arm died within the study period, which was 29 days. And that's compared to 15.2% in the control group, so placebo group. This gives you a hazard ratio of 0.73, so it looks less, but it's not quite statistically significant. So this would be called a trend. And the reason you see it's not statistically significant is because the confidence interval, which I've bolded here from 0.52 to 1.03, overlaps 1. And that means that it could be, there's no statistical support to say that it's definitely away from 1. So, fast forward to this week. So there's this paper, the article from the New York Times, which was published on October the 15th and updated today. The title is Remdesivir Fails to Prevent COVID-19 Deaths in a Huge Trial. Critics say the study sponsored by the WHO was too poorly conducted to be definitive. So on face value, we have two conflicting results. There's one study that was run, funded primarily by the United States government, that was run in the spring, says remdesivir is a useful drug. And here are the headlines that I'm sure you've seen this week, not just from the New York Times, pretty much from everybody else, that this trial claims that there's no effect. So, and this is a very common situation. I mean, science is, as a process, a lot of results, have to be verified. A very number of them don't get verified and they have to be replaced or updated. So what can explain apparently different results? Here are just some of the possibilities. So one possibility, and this is sort of my prior when it comes to press coverage, is that the results are not different, they're just misinterpreted. And we can try to decide if that's the case here. Another very plausible explanation is that clinical trials are basically statistical experiments. So there's some noise and some possibility that an experiment will find a result where there is none. So you could have a margin of error. It could just be that the original trial just happened to be one of the lucky draws and the replication failed. You could have a number of ways in which two trials are not directly comparable. So the fact that they're getting different results might be not due to remdesivir, or the action of any particular drug, but due to other factors. For example, systematic differences in trial designs or outcomes. They could have simply measured different things. They could have tested different drug combinations. It is entirely possible, although not very likely, that one or both trials actually made analytical errors, or there was some overseen confounder. There could also be a systematic difference in study populations. So perhaps remdesivir is effective in one population, but not in the other. And finally, it could be the fact the results are actually different. So one study is wrong and one study is right. So first, let's see what the other study says. So the first thing I would like to note, and I started by describing various sources of information for COVID or any other medical or scientific information in lecture one. The only record on the WHO trial at the moment, called the WHO Solidarity Trial, is available as a medRxiv preprint. So this is not, one, it's not the final version. They might have updated it with new information. And two, it has not been peer reviewed. So this is basically potentially subject to revision. So now let's sort of look into what this study actually did. So if you recall, the AGCC trial looked at one drug, and here is sort of an omnibus trial. They've looked at 4 different drug combinations. So looked at remdesivir, hydroxychloroquine, lopinavir, fixed dose combination with ritonavir, which are protease inhibitors from HIV, and interferon beta 1A. The trial had what's called an adaptive design, which means that unpromising drugs could be dropped and other added. The COVID-19 patients were randomized equally between whichever study drugs were locally available, an open control, up to 5 options. So the primary analysis were of in-hospital mortality in the 4 pairwise comparisons to each study drug versus its controls. And then they basically looked at survival for aggregated populations, and that's not entirely true. So there's, when you sort of, if you do the side-by-side comparison of what this trial did and the AGCC trial, or no, what was it? I forgot the abbreviation. Let me look at it. ACCT1 trial did. There are actually enough technical differences that you could potentially, explain any differences away there. It is a much larger trial, however. So one is 405 hospitals, 30 countries, 11,266 patients. So just the raw numbers are quite about 5 times larger than the ACTT trial. So 2,750 were allocated remdesivir, 956 hydroxychloroquine, and so on. About 4,100 had no study drugs, so they were in the control. It was reasonably good compliance, so that means how many people actually stayed in a treatment. There was a statistically large number of deaths reported, so 1,253. Median day after hospital admission was 8. And then the 28-day mortality rate was 12%. So if you compare it to the ACTT trial, it's the same ballpark. So there it was, either 11 on remdesivir or 15 without. And that was significantly higher if the patient was already ventilated in randomization. So this is a predictor of poor outcome if you need 10% otherwise. Now, this is sort of the key results of the paper, which is, what did each of the drugs did. And there's a lot of abbreviations and high density information recorded here. And we've seen this before, but let's just go through it one more time. There are 4 different drug combinations being treated. So you report first the relative risk of 0.95. So remember, this is the risk of dying if you get remdesivir versus the risk of dying if you don't. A successful drug should lower the relative risk dramatically. You can see the raw numbers. So there were 301 deaths out of 2,743 subjects that received remdesivir. And there were 303 deaths in the arm where there was nothing. So basically, we're talking about, 2 fewer deaths. The relative risk is very close to 1. The p-value is insignificant, so there's no effect that they measured for remdesivir. In this particular study, hydroxychloroquine actually increased the relative risk. Again, it wasn't significant, but just a point estimate was higher. So giving it to this particular study group was actually harmful. Lopinavir had no effect. Interferon had a worse effect. So no study drug definitively reduced mortality, either in unventilated patients or any other subgroup of entry characteristics, initiation and ventilation or hospital duration. Now there's, this is a verbatim paragraph from the paper because it sort of highlights the qualitatively different nature of the study. If you remember, the ACTT trial was meticulously structured, right? So it was very, very carefully laid out, what was going to happen. Everything was blinded. It was, predefined everything. Here it was a lot more organic. So the protocol was designed to involve hundreds of potentially overstressed hospitals in dozens of countries. Hence, no form filling was required, and trial procedures were minimal but rigorous. Online randomization of consented patients took just a few minutes, as did online reporting of death in hospital or discharge alive, plus brief details of respiratory support. No other reporting was required unless doctors suspected of an unexpected serious adverse reaction. National and global monitors resolved queries and checked progress and data completeness. So there is, and I'm not actually attributing any significant differences to it because I don't have access to the protocols, but there's enough differences between these trials that if you wish, you could attribute any differences to the way the trials were designed and run. And you will see this is actually exactly what Gilead is doing. So eligible patients were adults over the age of 18, hospitalized with a diagnosis of COVID-19, not known to have received any study drug, but this is probably self-reported, which means that this is not 100% accurate, without anticipated transfer elsewhere within 72 hours so they could be followed, and in the physician's view with no contradictions to any study drug. Randomized in equal proportions between control and whichever other study drugs were locally available. Placebos were not used. And then remember, it was an adaptive trial, so they decided that hydroxychloroquine and lopinavir were discontinued for futility, meaning it was determined based on the preliminary analysis that we're not doing anything, so they stopped it, and interferon is ceasing on October 16th. There were also 2 different primary and secondary endpoints. So what it is the trial measured: assess effects on in-hospital mortality. In other words, mortality during the original episode of hospitalization, follow-up ceased at discharge. Now, which is kind of an important point because potentially this person could be discharged from one hospital, then be admitted to another hospital and die, and that would not be recorded. Not only in old patients, but also in those with moderate COVID and in those with severe COVID, subsequently defined as ventilated when randomized. And then secondary initiation of ventilation and hospital duration. Yeah, there's just general statistical analysis. Here's the table 1, which describes the study population. So if you remember, one of the things I told you about when you scan these tables is, 1, you want to see which populations are being represented. So they will report all of the important covariates. So how old were the individuals? How severe was their condition when they're admitted? Did they have bilateral lung lesions? Basically, did they have pneumonia? How much time they spent in the hospital prior to this? Where it was? And other risk factors. So things like, you remember, being male is a risk factor for COVID, current smoking, history of diabetes, and so on. So you want to see if it's representative. And second, what you want to do is you want to see that there's no systematic bias between case and control. So you can kind of scan down the line and make sure that these are kind of balanced, right? So there's not active and control are not overloaded for too young, too old, and that all checks out. This is also a standard flow, so you can follow how this was done. We don't really care about hydroxychloroquine. So we can only look at this arm. So you had 2,750 active remdesivir, and then a matched number for control. These individuals dropped out because they failed to, basically didn't give consent, and here's the resulting population. And as should also be familiar by now, these are survival curves. which show you so, what was being shown here is remdesivir in control. And this is mortality. And this is the same thing, just, the inset is just not on a 0 to 100 scale, it's just blown up. So you can see the pattern more closely. But basically, there's no difference in survival. And just to remind you what this is that if you look at a particular time point, it tells you what fraction of all the individuals in the control or remdesivir arm died by that point. So, by 7 days, looks like about, well, we can look at this, 5% died. By day 14, 10% died, and then kind of tapers off. And by day 28, you have, what about 12%? But the key point is that these curves never separate. So from this, if this is your primary outcome, there's no evidence that giving somebody remdesivir improves their survival chances. And just to scan others, as I mentioned for hydroxychloroquine, there was also a statistically insignificant result. So that's how you should read it. The statistical test tells you there's no detectable difference, even though it looks like the rate of death is actually higher for hydroxychloroquine, but it's not sufficiently different, and the same for others. And here's another chart that shows you the same information, but just sort of in terms of risk ratios. So this is ratio of death rates, and 1 means no effect. Greater than 1 means that you actually have worse than giving somebody drug worsens their outcome. And these bars, so the whiskers around the square, are the confidence intervals. So if they overlap 1, there's no difference. Right? You can just scan down the line. Basically, every single measurement overlapped once. So this trial says there's no power to say that anything is effective. And this is the same thing broken down by whether or not, but by patient characteristics. And they also compared to the ACCT trial, which showed that remdesivir was particularly effective in the low flow oxygen 2 cohort. So this is, but in this, they actually didn't separate them out in this study, so they're not directly comparable. Now, if you go back to the New York Times article that I started with, here's sort of, the way they do a write-up is they go and, contact a number of experts. That's pretty random. I mean, I've been contacted to comment and things like this. So there's a statement from me on one of these things and in the New York Times from April, but it doesn't really matter. So they're kind of randomly selected by who the journalists contact in response to them. These people will typically not be, well, actually not typically, but will always be people not involved with the study. And if they are, it'll be explicitly stated. So you want to get quote unquote independent opinion. Just to give you some quotes, conducted in dozens of countries with various healthcare systems and inconsistent treatment protocols, the data are difficult to analyze and compare, said Dr. Peter Chin Hong, an infectious disease expert at the University of California, San Francisco. Gilead disputed the conclusions of the WHO study, noting the variety of drugs and drug combinations had been evaluated under a wide range of circumstances and that more rigorous studies had found a benefit. Quote, consequently, it is unclear if any conclusive findings can be drawn from the study results, end quote, the company said in a prepared statement. No surprise there. Remember, Gilead makes $3,200 on each course of remdesivir treatment, so they have a very clear vested interest, and they will be the last to admit if the drug is ineffective in this setting. Dr. Andrei Khalil, a principal investigator of the federal study of remdesivir and an infectious disease expert at the University of Nebraska, so this guy is sort of the opposition, faulted the WHO trial for not having a placebo group and for allowing patients and doctors to know which treatments were administered. So-called open-label trials can skew the reporting of results. And finally, as for remdesivir, I don't think the study is the nail in the coffin, said Dr. Tayson Bell, a critical care physician at the University of Virginia, but I do think it shows that we have to be selective about the patient population we use it in. So, of course, the critical care physician has sort of the most apropos take, because in the absence of alternative treatments, you are going to give people something if you think even a slight chance of improvement is there, but you don't give it to people for which the studies have consistently shown that there's no benefit. There's a question, are there people who usually drop out of treatment considered as part of the overall sample size? It's a good question. So if you look at here, let me actually go back to the chart where this is shown. So they follow you they show you the entire chain of custody, so to speak. So you start with 2750 started, but they actually do 2743 versus 2708. So they do the final analysis on a subset of those N. I mean, they can't do study on people that basically said, you know, I'm not giving you consent to analyze my my information. All right, so this should, you know, give you some food for thought, right? Because if there's one thing I hope you learn from this course is to basically be able to look at the primary information and make these kinds of decisions for yourself, because that's pretty much the only way you can convince yourself that there's no sort of noise in the system. So what I'm going to transition to next is you know, two papers that give us an alternative, because at the moment there are no specific target designed or custom, you know, drugs designed specifically for COVID, so for SARS-CoV-2. Another question, maybe very unrelated, but in the news, there was a company that paused trials for a vaccine. Do we know why? I think it was the AstraZeneca Oxford trial. They had an adverse effect, right? So somebody had an unusually severe reaction, which according to the trial rules, you know, requires a pause and evaluation. We will not know why immediately, because this basically is, think about this as a private health information, right? I mean, but revealing what happened would pretty much amount to describing what happened to a specific individual and their personal medical history. It's not necessarily HIPAA. So HIPAA is in the United States, it's in the UK, but they have different rules, right? I mean, if the trials run, but they're running multiple trials. But you know, yeah, there's a lot in the news. Like there was today a news item that I saw out of the corner of my eye. This was actually not. It's alleged that the adverse event actually occurred in the placebo arm, so it might have nothing to do with the vaccine, but it's unconfirmed, and I don't know. But what we do know is that there was one adverse effect that was sufficiently severe to pause the trial. This is nothing unusual; it happens all the time. So we did not have a drug that's provably effective. At best, we have a drug that's marginally effective, and you know, only at shortening your hospital stay. It doesn't actually increase your odds of survival based on the current available data. So what can we do instead? I mean, we're waiting for custom drugs that are in the pipeline, but one of the things that was also in the news because of President Trump's bout with COVID, that he received Regeneron monoclonal antibodies. So I was going to spend a little bit of time talking about that, because this is sort of shortcutting the discovery process, because instead of developing drugs from scratch, you know, which takes a long time, you know, you develop compounds, you test them. What you can do is you can basically copy what nature does. And this is what Regeneron more or less did. They start with the premise that the most effective antiviral killer that we have is the immune system. The immune system kills viruses through development of neutralizing antibodies. So they're molecules that are natural virus killers. If we can identify specific antibodies that are highly effective against SARS-CoV-2, you know, we could package them, you could isolate those, purify those antibodies, basically productize them, and make identical copies of those molecules and just deliver them in appropriate packaging to the patients. We also have, and they don't have to be developed from scratch, so you don't actually have to engineer it. What you do is you basically run infectious systems and see what the bodies do. And what Regeneron did is they've taken two streams of sort of antibody producing systems. So they looked at humanized mice, which are basically mice that are genetically modified to be immunologically identical to humans, and convalescent sera, so blood from individuals that had COVID and recovered, for quote unquote effective antibodies. And they wanted to isolate, purify, and characterize the antibodies. And the focus of the paper was to, instead of look at cross-neutralizers, so if remember, cross-neutralizers would be antibodies that are effective against multiple coronaviruses, so SARS-CoV-2, SARS-CoV-1, maybe something else. And we looked at papers where this was described. Instead, they went for antibodies that bind to a specific SARS-CoV-2 spike with very, very high efficiency. So they measure this in terms of concentration. So this is picomolar concentration. And the lower the concentration is, the more effective the antibody is, because you need less of it to neutralize the virus. So you have 7 to 99 picomolar concentration. And all of these neutralizing monoclonal antibodies bind to the receptor binding domain of SARS-CoV-2 spike and block its ability to interact with ACE2. So they are sort of the canonical way to stop infection. You clog up the virus, so it's not able to bind to the cell, which means the infection never occurs. And this is pretty much exactly what you want sterilizing immunity to be like. So here's, so they've identified a number of monoclonal antibodies. So each one of those is, so the next 4 slides are basically just confirming that these antibodies do what a Regeneron wants them to do, which is to kill SARS-CoV-2 or prevent it from infecting cells. And they do it with, in sort of 4 successively more realistic experiments. So you will see the same type of curves in all of these. So on the y-axis is basically neutralization efficiency, which is, what fraction of infection is stopped, right? So 100% means everything is stopped. 0 means it's completely ineffective. And on the x-axis, you have concentrations. So how much antibody do you wash in? And this is in, so picomolar would be 10. So 10 to the 9 is nano, 10 to the minus 12 is picomolar, right? This is millimolar. No, this is micromolar, and then millimolar is -3. So the less, so if you go closer to the origin in this plot, this means less and less concentration. So the most potent antibodies are the ones that rise the fastest. So here, for example, this, Regeneron 10-9-8-9, it's the first line that go up, and it basically reaches 100% neutralization at below nanomolar concentration. And what you have here, you also have 2 controls. You have IgG isotype control, which is just generic immunoglobulin. Generic immunoglobulin has nothing to do with specificity for spike. So it's just there to make sure your experiment isn't just binding everything. And this is the specific anti-ACE2 antibody, right? So this is known to inhibit binding to ACE2. So you see all the naturally developed antibodies are actually better than this anti-human ACE2 inhibitor. But this was, this experiment was conducted in a sort of in the least realistic system. So they used anti-spike monoclonals, IgG isotype controller, recombinant dimeric ACE2 were added to vero cells. So it's a cell line, that's monkey cell line. And some expression was measured after 24 hours after infectious readout for virus infectivity. So this is just one cycle in vitro assay. This is the same type of plot in a subset of data sets, except it's now in a more realistic system. They're using a different cell line and are using this VSV, which is a vector virus. I'll show you in a couple of slides what this is. But it also shows the same behavior. Now they're doing neutralization potency in against replicating VSV SARS-CoV-2 in vero cells. So this is a sophisticated in vitro experiment. We basically have a virus that has the exact SARS-CoV-2 spike engineered into it in monkey cells. The cells were infected with a multiplicity of infection. The virus and stained for viral proteins 24 hours after infection to measure infectivity. Again, the same. And this is actual SARS-CoV-2 virus, right? So this is not a construct. This is in cell lines, but this is now against the actual virus. And one of the things that they're doing on the last plot here, which is interesting, sorry, the last 2, is they're, in addition to using single antibodies, they're using these cocktails of 2 antibodies mixed together. Now what's shown here is uh, uh, it, you might remember, or you might not, we talked about immune antibodies have a particular structure. So antibodies have two chains. They have light chains and you have heavy chains. At a light chain and a heavy chain, each basically get to pick one of the genes in the germline. So you can think of this grid as a subset of all the possibilities that antibodies could pick, right? So your random antibody is going to pick one IgHV and one IgK, and then combine them together. But this is just sort of a categorization of which genes are being used by those antibodies. And the interesting thing here is the couple of things that are interesting here is that a lot of the antibodies that they found, they're basically variations in the same theme. So for example, there are 23 mouse-derived antibodies that combine IgV 353 and IgKV 1-9, but there's a variety there. So they're very different antibodies molecularly speaking, so that were developed through entirely different pathways, but they arrive at the same neutralization phenotype, which means that the immune system has many, many different ways to develop antibodies. And this is good because you want different antibodies that target different parts of the virus. And you probably know why, because of the assigned paper that you have to read and do a quiz on. And this is what they did here. They did a functional screen to try to see which parts so this is a sort of a really low resolution 3-dimensional structure of spike, and different colors are patches on the spike where the antibody can bind. And this is a view from the front, from the top, and from the back. So, and if you're looking at where these antibodies bind, so for instance, look at this guy. So Regeneron 10987. The color code is basically where it binds. So white means it doesn't bind there at all, and then colors mean how well does it bind, so strong or weak. So this antibody binds basically to this pocket. This antibody binds to a bunch of different pockets, including this one. The only thing you really want to take away from this is that all the antibodies seem to bind to slightly different parts of the virus, which is promising because they're hitting different targets. There's a question: when making a cocktail of antibodies is specifically made so that none of them overlap and compete with each other, or can you just have all of them together anyways? Great question. You ideally want to have as few of them as possible to target as many different things as possible, right? And you want to have as few of them as possible because, one, it's easier to make, it's easier to deliver, and there's less opportunity for interference, and you want them to target different targets so there's no evolutionary escape. So there's a high resolution picture of where they bind. So this is a cryo-EM map. By the way, there was an unrelated paper that just came out in Nature. So cryo-EM is this high throughput crystallization technique where you basically do electron microscopy on super cooled molecules, so you stop their motion. And an angstrom resolution, which is atomic level resolution, is what they've been trying to do for a number of years, and they finally hit it. So there was a break. We'll get better structure, even better structures moving forward. But what we have here is the part of the virus that the antibodies target is blue. And you have Regeneron, 2 antibodies shown here, Regeneron 10933, the green and cyan, so it's this body, this antibody, and the other antibody is here. And they see they hit the receptor binding domain in different spots from different sides. Which means that if the virus somehow manages to develop a mutation that prevents this antibody from binding, the other antibody is still going to come and get it. So the result of this paper was they generated a large collection of potent and diverse antibodies to meet the prospective goals of identifying highly potent individual antibodies, which could be combined into a therapeutic cocktail. Inclusion of such antibodies into a cocktail will deliver optimal antiviral potency while this got cut off, minimizing drug resistance. And they're actually being trialed right now. So an antibody cocktail is now being tested at this clinical trial, 2 specific clinical trials. And, as we know, Trump received one of them as well. So he was also a, clinical trial subject, size of 1. And based on him, it's a complete success, but who knows? So moving on to the second study, which you all read, so I'm just going to go through it quickly, is now that they have multiple potential agents, they want to demonstrate experimentally that you really need more than one. So as the previous study showed, promising monoclonals have been isolated from humanized mice in convalescent sera. Which is, if you remember, again, it's just, different recovered individuals. Whenever you have an antiviral treatment, drug resistance due to the rapid mutation of viral pathogens is always a concern. Resistance becomes faster when selective pressure is applied in the setting of drug treatment. The canonical example is HIV drugs. Just as a brief history, HIV was originally characterized in 1983. The first drugs came on the market, I believe, in 1987 or 1988, AZT. There were a few others. But the successful therapy did not enter the market until 1995, 1996, when there were were several drugs that are targeting different molecules in HIV. And those initial drugs are AZT, even though they would basically completely kill the viral replication for 2 weeks, the virus would always deterministically escape them because it wasn't completely effective at eliminating it. It just kind of knocked it down. And then because all you would need is one mutation, the remaining virus would generate this mutation and grow out. The reason combination therapy is successful is because it requires the virus to simultaneously mutate at multiple genetic positions. And this is an important idea. This is actually very, very true. This is true in viral therapy. This is true in bacterial and antimicrobial therapy. This is also true in cancer because monotherapies are not successful for aggressive cancers. So one of the things that they screened for here, this is kind of a paper that's short but dense, is so the reason they did this analysis is in the first paper where they were discovering antibodies, they only looked at the wild type. So everything was raised against the wild type virus. And this is what they measured. And this is what the antibodies were designed to target. So here you can see the wild type is what was originally designed to do. And these are picomolar concentrations. So the lower, the better. And all of these variants are this wild type with this one change in spike. And this denotes, for example, that V341I means that the wild type has a valine, an amino acid valine in position 341, and a mutant as an isoleucine. The reason they pick these mutations is because they're actually observed in circulating population. We have something like 130,000 sequences from different countries in different times. We can sequence them, we can look at them, we can find all the frequent mutations. If you look at this table, which is actually a really poor way to present the data because we don't, this is not something that we're good at recognizing as humans. I should have done colors or something like this so you could just immediately see that all of these antibodies are highly effective against each of these variants, right? So if you gave it, basically if you took this antibody and gave it to anyone that is likely to be infected with a circulating virus, regardless of which mutations they carry in the receptor binding domain, those antibodies would be successful. So that's good. There's no pre-existing resistance. Now they've done a simple passaging experiment, and this is a very, very this sort of about as simple experimental evolution as you can do. So first, you actually do selection not on SARS-CoV-2 because you need biosafety level 3, and there are other concerns there, just difficult to work with. But you take this VSV virus, so these NPM and L are proteins from VSV, and then you engineer it to have the SARS-CoV-2 spike. So this is basically a different virus that is modified to carry this, the spike protein that you want. So then what you do is you take Vero cells, infect them with this virus, and then incubate them in the different antibody concentrations, right? So the idea here is that with no antibodies, just control, very high levels of antibody should completely inhibit viral replication. So nothing gets through, nothing gets, but if you make it sort of very, very dilute, there's not enough antibody to stop all the replication, but there's just enough antibody to kill the viruses that are susceptible to it, that what's left might be resistant. So, and the reason for it is that you infect multiple cells in each one of those, then the virus will mutate randomly. You see which ones grow out in the low concentration, and then you passage them again, you grow them out at higher concentration. So you're basically stepping up the selective pressure. So here, you give it just enough antibody to promote the viruses that have an escape mutation, but not enough to kill everything. Then you select those viruses, and you passage them again, and then sequence what's there. This is sort of the summary result. So they're passage 1 and passage 2. They had isotype control, which means there should have been no effective antibody. As you can see, everything grew out. Here you have different concentrations of antibody, and this is the fraction of wells that had a reaction, right? So greater than 90% means basically everything grew out. 0, nothing was detectable. 20% is 1/5, which is and this is what they select for the second. So you can see that for the first, if you get monoclonals, just one of these 4, up to concentration 0.4, everything grows out, and they're able to basically, there's enough virus that escapes the action of this monoclonal antibody at this concentration. For 2 of the 3 combinations, this doesn't occur. So this highly diluted concentration is enough to stop viral replication completely. So they have to go down to a lower concentration. And then in passage 2, what they've done, they basically selected viruses that already had some escape mutations, and they're much more successful at defeating the antibody. So for example, here, Regeneron 10-933, the original population, only 20% grew out at 0.4 and like 5% at 2 and nothing at 10 or 50. But if you select viruses with mutations, now basically at 0.42, they're now 100% grow out, so that they escape the action, and they've actually almost escaped even the highest concentration of the drug. But for these 2 combinations, 10-898 and 10-897, they were not able to grow out at all. So the assumption is that the virus is unable, at least in this experimental setup, to quickly develop escape mutations. Then what they did is they took those outgrowth viruses and did high throughput sequencing on them, and just measured which mutations had variants in them. So this is a passage 1 and passage 2. The blue part is where you have a receptor binding domain. So this is what the antibodies target. There are a couple of interesting and sort of subtle observations you can make. One is that the virus mutates anyway, with or without the presence of an antibody. So if you look at this position 655, basically in every single measurement, with a few exceptions, there's a large fraction of the virus that developed this H to Y mutation. But it is not at all related to the antibody, because notice that it occurs even in the experiment where there is no antibody at all. It's just the virus. So this just might be a mutagenic position. The same at 682. So this could just be a part of, say, passaging into vero cells or something else. So these mutations are relevant to antibody escape. The ones that are relevant are those that, 1, do not appear in controls. So they should be 0 in isotype and virus. And 2, the ones that appear in the receptor binding domain, which is what, based on the previous paper, the monoclonal antibodies target. You can see they all really like this 484 position. And there's sort of a scattering of escape mutations depending on which antibody they go for. So this was just, they observed this in VSV data. So then they went on and checked that these mutations actually confer escape. So they took wild type and they engineered 1 mutation at a time. And you can see, for example, with Regeneron 10-989, engineering any one of those mutations, 4 mutations, 484, 485, 486, 493, completely destroys the ability of the antibody to neutralize the virus. So any one of those mutations will do. Any one of those 2 mutations will do for 10-987 and so on. For combinations, none of them, for these 2 combinations, no individual mutation has any effect on neutralization, which is sort of probably the cleanest experimental confirmation that one, viruses escape very quickly, and two, combination therapy is necessary and effective. All right, let me look at the questions. There are two questions. Does the type of mutation make anything different? Yes. So, I mean, in this, the thinking is the antibody binds something in the receptor binding domain, and some of those, sometimes it might be enough to change just one residue to break it up, and you can see it here, right? But it's not enough to change a random residue. You have to change a specific residue. The reason this gets selected is because, remember, these RNA viruses have a very, very high mutation rate. So if you take, say, 1 million or 10 million viral particles, infect the cell culture with them, they're all going to generate random mutations. But by chance, a fair number of them are going to generate the right mutation. Then you select those because those are the ones that grow out and put them in pathogen culture again, so you amplify this population. This is how you see it here. And the idea is that it has to be in the receptor binding domain because that's what we're targeting with antibodies. Second question, are alternative splicing events a possibility in coronavirus replication? Alternative splicing, no, but recombination is possible, right? So alternative splicing has to do with, you basically need to have exons and introns because you arrange genes differently. In virus, something like this could happen. I mean, like if you remember in SARS-CoV-2, you have the slippage site, so you have alternative translation, but it's mutagenesis, just a single point mutation, is much more likely to be the primary mechanism. Remember, thankfully, viruses are very, very simple genetically. They don't have very many genes. They don't have long-range regulatory complications. That's good for us because we can study them and we can see where it goes. All right, so I'm going to switch gears and talk about something other than coronavirus, but it has because I'm sure you're probably tired of it. But there's a lead in. The drug resistance mutation is something that is a thread that I'm going to go carry through into the second part of the lecture. And what I want to talk about is the second or first leading cause of death in the United States, which we kind of forgot about in 2020, and that is cancer. So first, if you've never looked at this resource, so crcancer.gov, it's an amazing resource for all kinds of statistics, very, very detailed about cancer in the United States. So I'll start with a couple of facts just to sort of frame it. So in 2020, it is estimated that 606,520 people in the United States will die of cancer. This is just an average cancer year, right? Compared to SARS-CoV-2, at the moment, we have over 200,000 deaths, give or take. So, let's say worst case, 300,000 deaths in 2020. This is a very, very, very unusual year for infectious disease for obvious reasons. A common, completely average year, cancer, twice as many deaths, right? So, and heart disease is another one that is very, very common. So this is the second leading cause of death in the United States. About 1.8 million people will be diagnosed with cancer in the United States. The rate is about 442.4 per 100,000. The rate of death is 155 deaths per 100,000 per year, age adjusted. Another interesting and perhaps striking fact that if you're alive today, there's a 4 in 10 chance that in your lifetime you will be diagnosed with some kind of cancer. So this is probably a striking statistic. And in 2017, there were an estimated just under 16 million people living with cancer of any site in the United States. And by any site, it doesn't mean location in the United States. It means which part of the body cancer affects. They also, Sierra also has great trends in terms of what happens to cancer over time. So there are a couple of things that are apparent. Is that over the last 30 years, both the rate of new cases and importantly the death rate, and this is all normalized per 100,000 people, there have been declining. And at the moment, the 5 rate relative survival is 2 out of 3. right? And this is based on lagging data, so from 2010 to 2016. And this number has been steadily going up. If you, again, sort of digging through what SEER has, you could break down all cancers by the most common cancers, right? And this is ranked by how many new cases there are, right? So breast cancer female is the most common, followed by lung cancer, followed by prostate cancer, followed by colorectal cancer, melanoma of the skin, bladder, non-Hodgkin lymphoma, kidney and renal, uterine, and leukemia. One of the things that's already obvious from this is that different cancers have very different death rates. So for instance, breast cancer, even though it has the most new cases, it is far from being the deadliest. So it's only 42,000, I shouldn't say only, but compared to the other cancers, this is a relatively small number. So for example, with lung cancer, the death rate is greater than 50%. Whereas, even though, I mean, it's not one-to-one, but 135 deaths attributed to this. Melanoma of the skin, on the other hand, is a fairly benign cancer, especially compared to lung cancer. Colorectal cancer is pretty bad, and so on. Question. So when we're considering cancer-caused death, are we considering it as the primary cause of morbidity? Yeah, so cancer-caused death is, death because of cancer. I mean, there could always be other things, but there are rules about how deaths are recorded and attributed. So this is the official cause of death, as recorded by the physician or whoever certifies the death certificate. Cancer is also not. It's sort of historically been called a disease of old age. How bad do environmental factors contribute to all these cancer cases and death? Wait a couple of slides, I'll get to it. It depends on the cancer, obviously, a lot. So the median age of diagnosis for cancer is 66, which means for most of you, this is a very, very distant possibility. I truly hope that none of you ever have to deal with this, but statistics at the moment say that some of us will, 40% throughout the lifetime. But there's a very skewed distribution of when an average cancer case is diagnosed. So there's a small number, about 1% of all the cases are in young people, and then it steadily goes up, actually peaks out at 65 to 74, and then it drops down again. So sort of this is the critical peak. If you make it over 60, if you make it over 75, you're probably going to die of something else compared to this band. So I'm right here. I'm in the 12.5%. So, I'm starting to feel the pressure. If you also break down the cancer cases by various demographic factors, so this is by race and ethnicity and male or female, you can see that the aggregate rate of men, and this is again all standardized by 100,000 persons. So men tend to get cancers more often than women. There's sort of a divergent and discordant pattern once you break it down by race. So for example, black men get cancers more frequently than white men, but black women get cancers less frequently than white women, and so on. So there's a difference in basically all of these categories, but it's not sort of super striking. Except perhaps for Asian and Pacific Islander, which is dramatically lower than, say, the whites. Let's see what the questions are. Looking down at survival rates, so at the top you have rate of new cases and death rate. So this is a zoomed in version of the picture that I showed in the first slide. And this is perhaps the most encouraging picture of all, is that the 5-rate relative survival is sort of trending up consistently. So in 1975, the 5-year survival rate was 50%, and now it's touching 70%. So it's, and importantly, this actually been a very non-uniform, so for some cancers it's much, much better than this. You can also get to very detailed profiles for individual states. So for example, here's the breakdown for Pennsylvania, where you could see, for instance, how does it compare to average American national rates. Again, this is not, there's no statistics associated with it, so you can only look at raw numbers. Raw numbers might indicate that Pennsylvania is slightly worse than the national average for most, but not all cancers. So for example, we do better for, marginally better for stomach and say prostate cancer. You can sort of scan this way. You can also break it down by how we're doing in terms of increase or decrease. So here are the cancers, and this is the 5-rate change incidence. All right, so how does 2017 compare to 2013 for all ages, both sexes and all races, including Hispanics? So you can see the best cancer in terms of falling incidence was prostate cancer, followed by melanoma of the skin and so on. And the worst ones is pancreatic cancer. And the ones with a hash, there are enough cases to say that this is a statistically significant difference. All right, so what are, and how's this relating to genomics? I would just say that cancer is perhaps the better case for involving genomics, both in terms of understanding. Firstly, cancer is a genomic disease. It is caused by mutation. Second, it's tends to be very different between people, so that's a reason for very personalized diagnostics. I'll show you a little bit towards the end why it is. And three, it's also a very, very promising way to, genomics is a very promising way to sort of catalog the changes that occur in cancer and try to see how we can fight them. Treatment today consists of old and tried techniques like surgery to remove the actual tumor, chemotherapy, which is designed to kill a lot of cells, but primarily target cancer cells, so it's a toxic therapy. Targeted therapy, which is designed to identify and home in specifically on a particular type of cancer cell. And this is potentially, where personalized medicine is the most promising. At the moment, and this is a little bit out of date. Some of these data are behind paywall, so this is about 2 years ago. Over 20 tumor types are being treated with new medicines that have been launched in the past 5 years. This is a very, very active area of pharmaceutical and biomedical research. So for example, in the period from 2011 to 2015, there have been 70 new oncology treatments launched for 20 different uses. As of 2015, half of the new active substances are available in 20 or more countries, and 12 new active substances are available primarily in the US. A lot of the early treatments are available in the US and in Western Europe. There's a lot of research being conducted. So here's an example of some of the new drugs. This is the newest, but this is recent. So we're talking about 5 years ago. So you can look at sort of this neat chart, and there are a couple of things that pop out. So you can see that there are several types of cancer that have a lot of new agents. So lymphoma is one, leukemia or blood cancer is another one, sarcoma, lung cancer, multiple myeloma. And there are a couple of cancers that basically don't have anything. So pancreatic, for instance. And there are a couple that only get, or a sort of small number of new agents. If you were to do an overlap, try to predict which cancers get a lot of therapies, a lot of these cancers in the top 10 or the top 20. And this is driven purely by the fact that, you want to treat the most common cases first, mostly because it's going to have the highest effect in terms of saving lives, and it's also going to make pharmaceutical companies the most money. Oncology research and development activity are tightly focused on targeted therapies. And this means that we're moving away from scorched earth, which a lot of early chemotherapy was, but to target specifically cancer cells. So small molecule protein kinase inhibitors, again, monoclonal antibodies, which you should recognize now, to block the cell processes that cause cancer cells to multiply. And the particular focus is being placed on targeted therapies that use gene marker tests to indicate a greater likelihood of tumor response, right? So this is the personalized medicine where you might have, say, breast cancer, but there are different genomic types of breast cancer, and there are particular therapies that are only effective at a particular genomic type of breast cancer. It has to be correctly diagnosed in order for the therapy to be effective. The late phase onco pipeline includes hundreds of biological therapies, broken down into things like gene therapies, monoclonal antibodies, or their analogs, potential vaccines for a wide variety of tumor types. And immunotherapies are one of the fastest growing areas with oncology R&D, which is, indicated in this slide here, where you can look at the change of where the pharma companies are focusing their energy. So targeted biologics has seen the highest increase from 2007 to 2017. Cytotoxics dropped, targeted small molecule dropped, and so on. So this is targeted biologics is basically highly specialized treatment for a specific type of cancer. Here's another way to look at it in terms of, and this, I think this is in terms of market, right? So if I remember correctly, the labels are missing, but this is in billions of dollars of where, and the relative fraction of the market for all of those, right? So this is also both growing and changing. Under what category would PARP inhibitors fall? I think that would be kinase inhibitors. It's a small molecule. But because they're targeting a specific pathway, I guess it would go into a targeted biologic. So oncology growth is expected to be in a very financially healthy, 7.5% to 10.5% range annually through this year, with global oncology costs will exceed $150 billion. Wider usage of new products, especially immunotherapies in developed markets who can afford it. Also, newer therapies with survival benefits will also bring longer therapy durations, offset by lower usage of existing treatments. So, sorry, the previous chart wasn't, let's see, what was it? Sorry, this is just the actual number, right? This is just the raw number of different different therapies. This is the market. And it's broken down by US, the top 5 European countries, Japan, far emerging, which is, I would imagine this is China, Brazil, the typical, the BRICS, and the rest of the world. As you can see, the lion's share of the market, where the expenses are, is still the US, Europe 5, and Japan, even though there's an increasing fraction of the pie that's going to these emerging countries. Now, let's step back from therapies and go back to the sort of basic question of, where do where do cancers come from? There was a paper, and I'll frame it by discussing this paper that appeared in Science a couple of years ago, with the title "Variation in Cancer Risk Among Tissues Can Be Explained by the Number of Stem Cell Divisions." Question, do you happen to know which European countries in the top 5? Not super important, but I'm definitely curious. I'm not 100% sure, but, because the UK was still a part of the European Union, I would imagine it's Germany, France, England, or the United Kingdom. And then from that point on, it's probably Italy. And then the 5th, I would, maybe Spain, but I'm not sure in the 5th. But, France, Germany, the UK, and Italy, I'm pretty sure are the top 4. But if we were to try to, guess it, it's probably based on the size of the GDP. So, whoever has the largest economic output. So here's a great, sort of the abstract. I really like this paper because it's sort of very simple, but it also carries a very compelling message. And it's sort of the type of paper where people made a connection that should have been obvious, but it somehow missed the attention of several generations of researchers. So the paper goes as follows: some tissue types give rise to human cancers millions of times more often than other tissue types. Although this has been recognized for more than a century, it has never been explained. Here we show that the lifetime risk of cancers of many different types is strongly correlated, so 0.81, with a total number of divisions of the normal self-renewing cells maintaining the tissue's homeostasis. These results suggest that only a third of the variation in cancer risk among tissues is attributable to environmental factors or inherited predispositions. The majority is due to quote-unquote bad luck, that is, random mutations arising during DNA replication in normal, non-cancerous stem cells. This is important not only for understanding the disease, but also for designing strategies to limit the mortality it causes. So, just your typical scientific abstracts are fairly, benign and, measured. But, they threw in this idea of bad luck, which then led to, a lot of fairly controversial press coverage. So if you do, you can do that Google search now. Obviously, this has all been superseded by the way COVID is covered. I mean, this is all really amateur hour. But this used to be an example of significant press coverage as scientific result. So, and the implication was, and then you had backlash, Green's bad luck cancer study and coverage, because it was interpreted, the implication was that if you tell people that it's bad luck and there's nothing they can do about it, then they will go on and do all the bad things that they're not supposed to do, like smoke and eat, drink and, sunbathe. But that's obviously not what the paper is saying at all. And let's see what it actually is saying. But yeah, the connection was made that people cannot, people will take this as leave to completely, forget prevention. But of course, that's not what the paper is saying. So in order to break it down a little bit, let's just sort of think about, what any disease factor, what the risk of any disease is composed of. We talked about this in the context of any disease in particular, any disease in general, and for cancer in particular, you can have a heritable component. So as you well know, there's certain germline mutations, for example, in carcinogen, in cancer driver genes like BRCA1, BRCA2 for breast cancer. So that would be one. You can have environmental, so exposures to carcinogens, and stochastic. All of these contribute. The contributions of all of those will be different for different types of cancers in different people, but what can we can we make some sort of broad conclusions? One of the things that is never been in dispute is that different tissues, among other things, have vastly different rates of cancer. So for example, if you look by where, you just take all the cancer cases, across the United States or the world, standardize them by, how many of them occur per lifetime per individual, and look at, which parts of the body they occur in. So 6.9% occur in the lung, about 1% in thyroid, 0.6% for brain and the rest of the nervous system. Then it keeps going down, 0.003% for pelvic bone, 0.00072% for laryngeal cartilage. If you look at this, this is 5 orders of magnitude, right? This is not due to environmental effects, unless I suppose you go and sit next to or inside the reactor for Chernobyl building and you get irradiated. But even then, you'll die from acute radiation poisoning before this. So this is not something that is just easily explainable by random variation. Question: stochastic refers to randomly generated somatic variants. Yes. Environmental risk, of course, is non-zero, and it could be very significant. So exposure to carcinogens or behavioral factors, for example, smoking, are all the major components of cancer risk, but they're also not 100% of the risk. So for example, if you just look at the stomach, right? So alimentary tract, esophagus, and the reason you want to look at this is the thinking is that if you have an exposure to a carcinogen in your intestines, all of them are going to be exposed to the same carcinogen. Like if you swallow not mercury, but if you eat something that is carcinogenic, all of these tissues are going to see it, perhaps a different, for different periods of time. But they're kind of a nice internal control system. So they're all affected by the same carcinogen, but nonetheless, you have a significant variation here. So esophagus is 0.5%, large intestine, which colorectal cancer is much, much higher, intestine and stomach much lower. So, there's got to be something else. It's not 100% environmental. And then cancers of the small intestinal epithelium are 3 times less common than brain tumors, even though small intestinal epithelial cells are exposed to much higher level of environmental mutagens that are cells within the brain, which are protected by the blood-brain barrier. Similarly, inherited risk factors only explain a small proportion of the variation by incidence, but they don't explain of everything. To quote from the paper, only 5 to 10% of cancers have a heritable component, even when heredity factors in predisposed individuals can be identified. The way in which these factors contribute to differences in cancer incidence among different organs is obscure. An inherited variant APC gene is responsible for both the predisposition to colorectal and small intestinal cancers, yet cancers occur much more commonly in the large intestine than the small intestine of these individuals, right? So same genotype, different outcomes in different tissues. So what's the difference? Let's look at the random component. And that takes us back to the 1950s, the genesis of the somatic theory of cancer. And it's a very simple theory that postulates that cancer is derived from a single somatic cell that has accumulated multiple consecutive mutations. The default state of cell proliferation in higher organisms, metazoa, is quiescence, so they don't do anything. Cancer is by necessity a genetic disease, so it's a disease that occurs when cell division generates mutations that control proliferation and the cell cycle, right? So not only is it a particular mutation, but it's a mutation that leads to runaway growth, right? And without it, you don't get your typical cancer phenotype. So the and this is sort of the original paper from which the somatic cancer theory, somatic cell cancer theory, sort of started. It's this paper from 1954 by 2 gentlemen named Armitage and Dole, the age distribution of cancer in a multi-stage theory of carcinogenesis. So what they did is they observed an empirical relationship, and I'll show you charts on the next slide. The cancer at a given age is proportional to the 6th power of the age. It's kind of a random expression, but let's see what that means. This agrees or comports well with the theory that you need 7 consecutive mutations that are needed to break a cell to a point where it sustains cancerous growth. The 7 consecutive mutations is very important because there's a chance of probably every time you have a cell replication, because they have to copy 3 gigabases of genomic information, there are going to be some mutations in that cell. But most of them don't actually succeed to grow, for multiple generations, where one mutation follows the other, follows the other, follows the other. And that is this subsequent order of mutations is essential. It's not just one mutation. You need to have one mutation, then another, then another. And this 7 consecutive mutations is sort of the break point on average. So, and the 7th order comes out like this. So the proportion of the probability of getting cancer, given that the current age is T, can be expressed, and this is a very simple model. You assume that PI is a mutation rate per unit time. This will depend on the genes. So the probability of getting these 7 mutations is the probability of getting 1 mutation, and the other, and the 3rd, and the 4th, and the 5th, and the 6th, and then the 7th. So you have this, you just rearrange this through simple algebra, and you get the product of individual mutation rates multiplied by the 6th power of time. This is where it comes out from, the 6th power of time. And this is all they did in the paper. They plotted, so if you take the previous expression and take the logarithm of both sides, you get that the logarithm of this probability is roughly 6 times the log of the time. So what they do here is they plot logarithm of age, right? So this is log base 10, presumably. So, log base 10 of 10 is 1, right? So this is 100 years, and this is, 20 years. And this is the log of death rate. And notice how beautifully they follow the straight line. And the slope is between 4.97 and 6.48 for colon cancer, men and women, or for rectal cancer in men and women. But there are deviations from the model as well. So not everything explains it. For example, for lung cancer, it follows quite nicely and then drops off. Follows quite nicely and then drops off. Prostate cancer is an entirely different behavior. So it's a sort of a much accelerated for older age. And this can be interpreted to take that individual and environmental factors modify risks in a time-dependent fashion, right? So, for example, the prostate cancer becomes significantly more, much higher risk for older men. So the bad luck hypothesis is also very simple. If the only time a mutation can arise, good or bad, is when you have a cell division, right? If there's no cell division, there's no opportunity for creating mutations, good or bad. So more cell divisions begets more risk of acquiring bad mutations. Only stem cells can undergo multiple rounds of passaging needed to have the opportunity to fix multiple mutations, because somatic cells die off, not enough. So look at stem cells. Once you get a bad cell, you're done for, right? So at this point, the cancer is deterministic. So again, it's just a model. And critically, it is assumed that the rate of somatic mutation in different tissue types is the same, right? So the only difference between them is how often do they replicate. Question is, but don't log graphs decrease the spread of the data? So if we plot just the rate of the spread of the data would be more. Well, yeah, I mean, it compresses the variation, but this is a valid, this is how you would fit. I mean, if you were to plot it in the other scale, you would have, just a much harder to interpret plot because you'd have a very, very steep curve, and it might not fit the points nearly as well. But you can quantify, how well the curve fits the data, with or without the log transformation. So this can all be done. That's a good question. Yes, doing log scale compresses the variation significantly. But what it usually does, it compresses higher variation. It's called a variance stabilizing transform, but let's not get into this. All right, so now we have enough information to sort of go back to the science paper. And this is an example where a graduate student working with a very senior cancer researcher actually did this paper because it was, so nicely framed. And they didn't have to generate any new data. So to investigate this hypothesis, basically the grad student did a very comprehensive literature search to find incidence of cancers by tissue and organ. This is this variable R. And then estimate how many, on average, stem cell divisions take place in this tissue or organ in the lifespan. So this is lifespan cell divisions. And then correlate the rate of cancer and the rate of cell division. And in order to count cell divisions, they adopted this formalism. So you take the number of cell divisions is proportional to the number of stem cells and the number of division cycles over the lifespan, because you want to count the number of cell divisions. So each cell gets to divide. There's an average steady state of these, so that you assume that this is a constant population. This is how often they divide. And 2 is because, you're diploid. So every time you get 2 copies. The most tedious part of the paper was to estimate how many cell divisions occur in a particular tissue type. And it's tedious, but it's, so they wanted to estimate, you have to count how many cells there are in a particular tissue type. So for example, the proportion of osteosarcomas in the legs are estimated this, blah, blah, blah. So you have to do these calculations and find how often stem cells divide. In the bone, they divide only once every 15 years, right? So in your average lifetime, they'll divide 5 times. And they did this, there's a supplementary table in case you're ever curious how often cells divide in your different parts of your body. This is a good paper to go look at it. So for example, here's, and this is alphabetical, and it's just a part of the table, but acute myeloid leukemia, this is lifetime cancer incidence. The total number of cells is, 3 trillion. Number of normal stem cells in tissue of origin, it's S. And then, so this is normal cells, normal stem cells, division of each stem cell per year, so 12, number of divisions in each lifetime, so assume 70 years. No, so 80 years. So 960 divisions, and this is the total number of divisions, right? So this is how often, and when you do this plot of total stem cell divisions by lifetime risk, you see a beautiful linear correlation. And again, this is a log-log plot. So you can see the common cancers like colorectal, they're both common and they occur in the tissue that divides a lot, right? And then you have like head osteosarcoma, arm osteosarcoma. So these are all bone cancers that are quite rare. And they also occur in tissues that rarely divide. And this is linear regression. Could a stem cell have cancer? Yeah. I mean, by design, this is basically what they're measuring here, right? This is stem cell divisions. I mean, any cell can have cancer, right? Because it basically just means differentiated or undifferentiated, just runaway growth. So R of 81, right? So this is the actual correlation. R squared is 0.65, highly significant. They put in some quantification because these back of the envelope calculations of how many stem cells there are were pretty inaccurate. So they saw how sensitive their analysis was. It was pretty robust, but you had varying degree of correlation. But roughly speaking, about 2/3 of the variance. So this is what comes from 2/3 of the entire variance in cancer incident is explained by stem cell divisions. A linear correlation equal to 0.04 suggests that 65% of the differences in cancer risk among different tissues can be explained by the total number of stem cell divisions in those tissues. Thus, the stochastic effects of DNA replication appear to be the major contributor to cancer in humans. There you go. So now you know where 2/3 of the cancers come from. This is just random effects, at least based on this paper. And this is all, of course, remember, when we're talking about this, these are all averages over averages, because this is talking about an average cancer, an average lifetime. There's always very large variance attached to it. So let's now look at the genomics of cancer and see where where some of the key concepts there are. Question: what about differentiating between carcinomas, blastomas, et cetera, or the phenotypic difference between these cancers? I'll give you a few examples later, but these are different. So traditionally, cancers would be you have solid and you have diffuse, right? Solid tumors, the ones that just literally grow as a lump, and then you have diffuse tumors like blood that just circulates through the body. I'm not by any means an oncologist. I know very little about the clinical aspects of cancer, only as much as I would as a consumer, a medical consumer, or if you have ever had relatives with cancer, you might learn about a particular type of cancer. But it's the traditional diagnosis of cancer is based on histopathology. So people will look at a specific cell and see how what's wrong with it, and the diagnosis depends on the cancer type. So it'll be, which cells are affected, and how are they affected. But now it's sort of this whole diagnostic hierarchy is also changing because of genomics. So genomics of cancer, they're sort of one of the early discoveries in genomics were motivated by cancer. It's been recognized in the late 20th century. So for example, the sort of gross genomic rearrangements, like the Philadelphia chromosome, characterized in 1973. It's responsible for chronic myelogenous leukemia. It's a large transposition where you have non-homologous recombination, right? So typically when you have recombination, you need two strands of chromosome 9 can cross over. What you have here is a non-homologous recombination where a part of chromosome 9 gets attached to chromosome 22, and it gets flipped around. And this creates what's shown here is this BCR-ABL. It's an abnormal fusion peptide. So it's a protein that's composed from an abnormal junction of things that should not be together. And this is what causes leukemia. And then in 1982, there was a point mutation in the gene called HRAS, and that was associated with bladder cancer. Now, we're in the age of high throughput sequencing. We talked about it in the context of SARS-CoV-2. There's really nothing different here, just different DNA gets sequenced. What it allows us to do is to directly measure genetic variability of cancer within an individual. When we do this, we sequence two sources of DNA. So when you sequence an individual with cancer, you need to sequence the normal DNA from this individual and then cancer DNA from this individual. The reason for this is because you cannot compare cancer from a specific genome to a reference from a database, because most of the difference are going to be normal. You need to have healthy DNA from an individual and cancer DNA from an individual to basically filter out all the normal variants and just keep the ones that are found in cancer cells. You can do all kinds of sequencing. You can do DNA sequencing, RNA sequencing, single cell sequencing. And I'll show you a little bit what we can learn from this. First of all, there's a tremendous amount of data, public and private. Private, who knows, because it's private, but there's, as far as public databases go, there's the National Cancer Genome Atlas, which is maintained by the National Cancer Institute. This is a flyer they have, and they still have it on their website, just a little bit out of date. So they have 2.5 petabytes of data, which describes 33 different tumor types from 11,000 patients, including 10 rare cancers. They have a public data portal, which you can actually go and get. Sometimes you might need to get to sign some usage forms or obtain approval. But you can go and look at what's publicly available. So for instance, at the moment, the public, this was from today, so about 85,000 cancer cases available there from 68 different types, which involves close to over 23,000 genes and catalogs over 3 million mutations. And here's the distribution of different cancer types. Again, you can see it's non-uniform. So, breast, bone marrow, colorectal, and lung, it's in thousands of cases. So you have, much higher representation, for example, compared to, say, thymus or testes or bile duct. The standard workflow for, cancer diagnostics using different using next generation sequencing of technologies. It consists in sort of these 4 categorical phases where you prepare the sample and do sequencing. So you always take normal tissue, which is important, and then you can take 1 or more different tumor tissues, sequence them, keeping them separate so you know which came from which, either by running separate experiments or by attaching tag molecules to them. Then the sequencer gives you a very large number, billions of reads, which you then assemble into cancer genomes, either by mapping to human genomes or doing de novo assembly. If you want to find some really strange rearrangements in cancer, then you reduce this to just variants. So where are the SNPs, what are the structural variations? And then you do analyses on just these differences. So the output of this analysis, you basically want to identify as few differential mutations or other changes between healthy and cancer cells as possible. And then you plug it through statistical analysis and do inference. So for instance, let me just give you a couple of broad scale review papers to show you what you can learn. So if you do whole genome sequencing, either just the genes through exome or whole genome sequencing, can help us identify cancer alterations by looking at different genomic scales. So we knew about, say very large scale rearrangements like the Philadelphia chromosome before next generation sequencing, and by specific genes for a while as well, but many have been discovered recently. So here's sort of the type of variations that people look for in cancer. And these are based on the size of the change. So this is the log scale, shows you how much of the genome is involved in this change. So point mutation is one base. Then you have microsatellite, which is a short pattern that repeats. So this is 10 bases. Then you can have an insertion or deletion where part of a gene, gets inserted or deleted. Then you can have a copy number variation where all of a sudden you have multiple genes with an abnormal number of copies. Translocation is, where a part of the chromosome moves around, and aneuploidy is where you have a whole chromosome copied over. So all these different scales. Question, so when you refer to cancer genomic analysis, do you always refer to analysis of a wide population? Can it be a case study for a rare cancer type? Typically, cancer genomic analysis is always done in a single patient. But then you can aggregate data over multiple patients to learn patterns across them. But the sort of the basic unit of measurement is actually just one cell within one patient. We can get to that point now where we can cancer. If you want to study, cancer, so different metastases or different tissue sources in the same patient, you can actually now get to the level of pulling out a specific cell and sequencing the genome from that cell and then combining that across multiple patients. But your sort of base unit of analysis is one patient. Now, the first kind of question we want is that I mentioned that the basis of the way cancer arises is by mutagenic breakage of a particular cell. And then, of course, once cancer starts going, these cells continue replicating at a much higher rate, so you can have more mutations arising. So the first type of question is, can we just sort of build a spectrum of how many mutations occur in a particular type of cancer? And this is just a survey study where you take a large number of samples from patients with the same cancer type, sequence them, and just count how many different mutations there are. And this is I think, a really neat plot that kind of shows you the different scales. Again, you should now be used to the log scale. So here on the y-axis, it's the number of somatic mutations per megabase, right? So here is 1 per million bases, 10 per million bases, and this is 1 per 100 million bases. There are multiple dots in each of the bars. Each of the bars is a particular type of cancer. So hepatoblastoma, AML, thyroid cancer, prostate adenocancer, and so on. Each dot is specific patient. The red is the median or the mean. I'm not sure, it doesn't matter. They're all sorted from the least mutagenic cancers to the most mutagenic cancers. There are a couple of patterns here that you see that should be apparent. Is in pediatric cancers, they tend to be much less mutagenic, so they're caused by just a few mutations, or they're associated with, and then with adult cancers, there's higher mutation rate. And there's, again, a dramatic difference in how mutagenic cancers are. So this is 3 orders of magnitude, almost 4 orders of magnitude. So this cancer has 1,000 times as many mutations as this cancer. So just based on this coarse characterization, you can immediately tell that there's something fundamentally different between them. I mean, how can you have a cancer where in individual cells you basically have almost 1 in 1,000 base pair mutations? I mean, this is like viral level of mutations. This is insane. So they differ by about 3 orders of magnitude. They're higher in adult cancers, potentially because of more cell divisions. And an interesting, there's actually some cancers that have 0 somatic mutations in exomes. So all the genes look intact, which means the mutations actually have to do with epigenetics or EQTL, so they're regulatory elements. So gene expression or gene silencing and activation. Now sort of to dig a little bit into why some of these differences occur is that the vast majority of common solid tumors have about 1 or 2 mutations per megabase. Now, tumors with much higher rates, so rates above 2, are typically caused by tissue exposure to environmental mutagens. So sort of broad scale DNA damage, so cigarette smoke or UV light, or ionizing radiation or what have you, some other chemicals. If you go higher than this, tumors with greater than 10 mutations per megabase, it means that the cancer itself broke DNA repair, right? So the cancer itself is now, it affected the genes that are involved in DNA copying. So for example, if you have this MLH1 gene, the cells that possess this gene are simply not able to fix mutations. So cancer cells keep accumulating mutations more and more. So MLH helps fix mistakes that are made when DNA are copied in preparation for cell division. And tumors that have over 100 mutations per megabase, they usually have DNA replication proteins that are damaged by cancer. Sort of a counterintuitive observation here that these highly mutagenized tumors sometimes have better prognosis. And suggested mechanism is that there's so many mutations, the cancer cells basically kill themselves by being too mutagenic. They're no longer able to survive and replicate. Simply, they experience something called mutational meltdown, which is why there's a limit on mutation rates. For example, this is why viruses cannot mutate faster. If you do that any faster, they will actually just die. Now, if you look at where these mutations are, there are also different patterns that arise. So in most cancers, cancer type mutations to a couple of key genes are found in a large fraction of the cancers. However, the tail of distribution contains several more genes that mutate a minority of tumors. Less frequent mutation in other genes could be basically breaking the same cellular pathway, targeting a novel pathway. However, and redundant mutations in key pathways are an increasingly common feature of cancer genomes. So by mutations in the pathway, I mean if this is a pathway that, for example, removes the brakes on cell division, there might be 3 proteins involved in this pathway, there's usually more, but you might have cancer mutations that affect several of those proteins at once. And part of the reason is because protein pathways are generally pretty stable, so they're able to tolerate some disruption. But if you have multiple disruptions, they will eventually break down. So this chart shows the distribution of which genes experience mutations in this one type of cancer now. So clear cell renal cell carcinoma, based on 417 individuals. And you can see that this is sort of a fairly canonical pattern where there's, say, 3 genes, VHL, PBRM1, and SETD2, and then maybe BAP1, that combined might occur in nearly every single case of this cancer. So if I want to design drug therapy for this particular cancer, and this is a repeatable pattern, I might start asking questions. Are these genes all doing the same thing? Are they all working together in the same pathway? And if they are, I'm going to dig into this pathway and try to identify, how it can block its action in cancerous cells. Cancers are like viruses, unfortunately, and that's because once you have this runaway replication process, they keep accumulating mutations. So you have intrahost somatic variability. Within an individual with sufficiently advanced cancer, you're actually going to have different cell lineages. So you're going to have cancers with different mutation compositions. So every patient is a different collection of somatic mutations. What this means, just like in SARS-CoV-2, just like in HIV, if you don't design a successful therapy that kills all of it, but only some of it, then whatever's left is going to become resistant. So for instance, colorectal tumor that are wild type for this KRAS gene, you can treat them with a particular type of antibody called anti-EGFR. However, this therapy almost always fails within several months of initiating therapy. And if you take the individuals that took this therapy and failed and sequence their genome, you will discover new KRAS mutations. Or it is also likely that KRAS mutations were already present in a small subpopulation of cells prior to the initiation of the therapy. You can model it and using population genetics and estimate that it will take only about half a year for a resistant subclone to expand and regrow into a tumor that is resistant to the anti-EGFR antibody. Which in this particular case means that, if you're treating colorectal tumors, you should target it with perhaps this drug, but other drugs as well. So it is no longer able to simply escape the action of that one drug. With NGS, if you find genes that are mutated in particular cancer, it is now possible to do a couple of things that would not be possible before. For instance, you can use drugs that are developed for a morphologically different type of cancer. For instance, you might use a breast cancer drug. I mean, just making it up, so it's not an actual example. But if it were targeting the same gene as a gene that's also implicated in skin cancer, you could do this. You could do cross-target treatments. Because cancers that look very different might, in the level of genetic modifications, have the same genes that are involved. And this is something that you could not discover just simply looking at cancers and what the cells are like. You would have to obtain high-resolution genomic data. You could also use NGS-based discovery to create a classification or a taxonomy of cancers based on which genes are mutated. So which pathways are being targeted by the drug, and not necessarily which organs are affected or how cells are pathological. And importantly, you can discover new genes to target for treatment. Here's some examples. For example, the new genes that were discovered through large-scale next-generation sequencing. And these are sort of broken down by broad pathway features. So for example, the pathway that ensures large-scale genome stability. So this POL gene basically regulates proper genome packaging and folding. And if it's changed, you can have colorectal and endometrial cancers. RNA splicing machinery, myodysplasia, chronic lymphocytic leukemia, metabolism, DNA methylation, chromatin compaction, transcription factors, ubiquination, tumor suppressors, RNA abundance, translation regulation. Notice that all of these are somehow involved in fairly broad processes that tend to involve some stage of DNA replication, gene expression, or in some case, tumor suppression genes. So for instance, if you just look at one, so whole genome exome sequencing identify that about 10% of endometrial cancer have an unusually high mutation frequency, so over 100 per megabases. When they then group them together to try to find which gene lights up repeatedly in those individuals, they were able to find somatic mutations in a specific domain, so exonuclease of the POL gene that has proofreading capability. Sorry, my description of the POL gene in the previous slide was wrong. It's a proofreading gene, not a genomic stability in the sense of structural stability, which is critical for high fidelity copying of the DNA template during cell division. Furthermore, 80% of all those mutations seem to hit two specific locations. And again, you should recognize this now from our discussion of monoclonals. This is a mutation from proline to an arginine at position 286, and this is valine to a leucine at position 411. Somatic POL mutations are also found in ultramutated colorectal cancers. And you could also say with high penetrance, if you have germline variants at those genes, 424 and 487, may be associated with susceptibility to colorectal cancer. And this is purely driven by large-scale genome analysis. RNA splicing. The mutations of various components involved in RNA splicing were first discovered in myelodysplastic syndrome, which in English means poorly formed or dysfunctional blood cells. Most mutations in this gene affect the 3'. So if you look on the, it's the right-hand side, as it's drawn, splice site recognition during pre-mRNA processing and induce abnormal RNA splicing and compromised hemipoiesis. And it's very common. So 45 to 85% of cases have mutations in the splicing pathway. Right. So this is again a systemic breakage. RNA splicing means your genes don't get assembled properly. Metabolic genes. This IDH gene, which you don't really think of them a priori that, why would you have metabolic genes involved in cancer? So they're not recognized as one of the most frequently mutated metabolic genes in human cancer. And then, of course, you want to propose the mechanism of how this works. These mutated isocitrate dehydrogenases lose their normal activity to produce this, alpha Kg. Again, a new activity producing a different molecule, D2-hydroxyglutarate, which is a structural analog of a alpha Kg, but is also a potent inhibitor of enzymes that depend on alpha Kg. And now we have the connection. These enzymes are called methyltransferases involved in DNA and chromatin methylation. And DNA chromatin methylation is involved in gene silencing or activation. So this is now basically becomes a mutation, dysregulates which genes are expressed and when. So it results in aberrant epigenetic modification, as well as the potential for deregulation of many other cellular pathways. So clinical applications are numerous. So diagnostics, if you have a drug, for example, that targets a specific gene, a specific mutation in the gene, you want to treat a person with cancer with that drug, you want to make sure that they actually have these mutations. And the other thing you can do is you can do cell-free, what's called liquid biopsies. So you can do DNA sequencing of cancer without having to do surgery or invasive removal of tissue. And that's because you have cell-free tumor DNA. It's about 5,000 genomes per milliliter of blood. So this is first very sensitive, and this allows you to basically do an exploratory discovery of potential cancer, without knowing what you might be looking for. Question: when we consider the function of inhibitors in cancer treatment, the reason why we don't use whole D1 inhibitors is because of non-specificity to cancer cells, present null cells. Even if inhibiting it could theoretically increase tumor mutation burden and promote cancer cell death. I'm not entirely sure. I mean, to answer this question, I would actually have to look at the specific therapy, but all of the concerns that you raise are important. So specificity and off target, and generally toxicity, right? You're not able to just inhibit critical functions of both healthy and cancer cells. The goal of a targeted therapy is that the ideal targeted therapy will go specifically to cancer cells and nothing else. And a lot of times it's really difficult to do, especially when you're looking, if you think about it, a lot of the proteins that are malformed are in parts of the cell that are very difficult to access by drug. They're in the intracellular cytoplasm. Some of them might be in the nucleus. So you have to deliver them there. And you have to deliver only to the cancer cells. And how do you know that it's a cancer cell? You look under a microscope, you can tell it. But if you're trying to develop a delivery mechanism, it has to have some feature on its cell surface. For example, it has to be express some receptors that are not present anywhere else. For some cancers, that's the case, but for some it isn't. So delivery and targeting is a huge, huge problem, even if you know what to target. So here's an example of how you might guide a personalized treatment of one of the types of cancer. So this is a busy figure, but I think it sort of shows all of the important concepts. So it shows a couple of different genes. So let's first look at the abbreviation. So these are receptor tyrosine kinase mutations in different cancers. So the KIT oncogene plays a role in testicular germ cell tumors, so this guy, as well as gastrointestinal stromal tumors, this guy, so primary and secondary mutation. So these are the profiles of mutations in different genes. This is one gene. This is the composition of the gene, where you have different exons. This is cell membrane. And what's shown here are the different mutations that can occur in each part of the gene. So, which part in this, so which exon it's in, which amino acid, and what's being changed. And this is the color code of different treatments you could try. So each one of those, IM, SU, SO, and IMI, and DA, they're different drugs. So here they are. So you have, IM is imatinib, SU is sunitinib, and so on. Depending on which mutations you have, some of these drugs will be more or less effective. So for instance, with a green circle, syndicated means that 5 of these agents don't care if you have a mutation in position 560. If you have a mutation in position 654, then IM is useless. It's highly resistant, but SU and SO are effective, and NI is sort of in between. So what this can already tell you is that, one, you look at which cancer it is, then you look at the complement of drugs, and then ideally, in order to determine whether or not you want to give an individual a particular combination of drugs, you need to sequence their genome. For instance, if they have, a cluster of mutations at exon 17, you can see that, IM and SU are completely useless. So you basically must give them something that includes this MI and maybe SO, depending on which mutations they have. And this is exactly, how you would use personalized treatment. You would design, and this is exactly how it's done for HIV, for example. If you're diagnosed with HIV, a doctor will order a drug resistance screen. So the lab will send them a profile of mutations in your virus that you can then match against which drugs are likely to work and which ones are not. And then you can tailor your therapy based on, what genomic profile the virus has. Obviously, this requires that you have multiple agents that have different specificities or different potencies against different mutations. Summarize next generation sequencing in cancer has given us an in-depth understanding of cancer biology, at the level of diagnostic, so what's there, prognostic, which is, which treatment is likely to work, and actionable biomarkers for cancer management. And then cancer-free DNA is a promising diagnostic biomarker, which I haven't talked about at all, but there are a lot of technical difficulties involved with it. There's a lot of very exciting developments in cancer treatment, but you can sort of start seeing how this goes. So you can target, instead of targeting a specific mutation, specific cancer, you might go for targeting core signaling pathways. So for instance, because cancers are very heterogeneous, hundreds of genes are involved, different mutations and variants, it is simply impossible to design a therapy that will target a specific combination of mutations. Even if you could do this, then think about the drug approval, right? I mean, if you have, how would you even do a clinical trial for a highly specialized treatment for which there may be only 10 people in the world that have this complement of mutations? So you got to do something else. The alternative is then basically go to a higher level, and instead of targeting a specific mutation, a specific gene, you could target a specific pathway, which means a molecular pathway which is involved in cancer's growth, so cell replication, cell division, formation of blood vessels, and all the sort of canonical things that are involved in cancer growth that also encompass all known genes. And then what you could do is you could basically say, in cancer, in particular cancers, my goal as a therapy designer is to disrupt the function of this pathway when it's dysregulated by cancer. And then you're free to choose which gene in this pathway you target. So for instance, KRAS, which a couple of slides ago I told you is something that's targeted by PKR therapy, but is able to, it's difficult to target directly. So instead you could focus on another gene in this pathway, which might be easier to target for mechanistic reasons. Maybe it's expressed earlier, maybe it's easier to access, maybe there's just a drug that's more effective against it. And for future treatment, it is also necessary to focus on combination therapy, both to combat resistance. And just like we started this lecture by talking about monoclonal antibodies for SARS-CoV-2, same exact principle here, except you have more targets. So there, everything was targeted the same protein. Here you have multiple proteins, so you could target different genes of proteins. You could also do targeted therapy plus immunotherapy. to inhibit essential molecular pathways that are required for tumor growth and survival. And complementary to it, you can apply immunotherapy that will basically recruit immune T cells to train them to kill tumor cells, which is both effective and long-term sustainable. All right, so that's all I got for cancer. Cancer biology and genomics in an hour. And for next time, since, let's see, we have, I think, 4 or 5 lectures left. I need to look, because after Thanksgiving, we're done. So let's say we have 4 lectures left. I want to make sure we cover all the preventions that have been tried, for SARS-CoV-2 and COVID, because that'll be a big portion of your final project. And there's a lot of incomplete data and controversy on a lot of them. So we'll, next time we'll start talking about non-medical interventions, and I'll have to put it in the context for you. So we'll talk a little bit about disease epidemiology. But read this. This is not a paper per se. It's a review. So it's called a news feature. It is, a more accessible review of other papers to describe, what other studies are saying about the effectiveness for face mask wearing for slowing down or potentially stopping SARS-CoV-2 transmission. Thank you all for sticking around, and I will see you